Um, so this is our next in the series of iPoLS seminars, monthly seminars, and today we have Daniel Witvliet and Vlad Susoy, who will be talking to us. Daniel is going first, and he will give his talk on the principles of brain circuit maturation by mapping synaptic wiring diagrams across development. And I think if you are ready, then go ahead and we're going to ask that y'all hold your questions until the end. And we would like students to ask questions before faculty, please. And feel free at the end to unmute yourself and just go ahead and ask your question. And with that, I think, Daniel, you may begin.
Great. Thanks for the introduction, and thanks for having us here today. Today I'm going to talk about a project that's been going on for many years across multiple different labs, where we've been trying to understand how the brain changes throughout life. Because the brain, which is the center for sensory processing, decision-making, and motor control, it has to be highly dynamic throughout life. And this is especially evident from birth to adulthood when the brain faces multiple challenges. Because during that time, the brain has to support new functions, such as walking and critical thinking. It has to maintain an already established repertoire of functions despite a constantly changing body. And to adapt and learn, the brain must continuously be optimized to a changing environment. And it's poorly understood how the brain copes with these challenges because of its vast complexity. So with billions of neurons interconnected by trillions of synapses, the human brain is simply too large to study all neurons in synchrony. Even smaller model organisms such as the mouse and the fruit fly still have millions, thousands of neurons. So to study the anatomy and function of all neurons throughout the entire brain, we have to turn to a smaller, much smaller animals such as C. elegans, the nematode, whose entire nervous system consists of only about 300 neurons. And even though the C. elegans nervous system is small, it's organized similar to the systems of larger animals, meaning that the worm has a brain where most sensory information converge and decision-making happens. It has a main nerve cord where much of locomotion is controlled, and it has peripheral neurons throughout the body, most of which collect information, sensory information that's directed into the brain. So because of the small size, we can actually map the entire nervous system at synapse resolution, meaning that we can get a complete diagram of how all neurons are connected to each other. We call this kind of neural wire diagram a connectome. Another advantage of the worm, other than it being small, is its stereotypic cell lineage, meaning that the birth time and location of each cell is almost identical across different animals. So we can directly compare every neuron, the number of neurons, but basically you can take any neuron in any animal and find it in another animal to see how its connections change from one animal to the next. And finally, C. elegans populations, they're isogenic as they make clones of themselves, so we can pretty much eliminate genetic differences as being the cause of any connectivity differences we might observe between animals. So altogether, this makes for an ideal system to extract fundamental principles about how and why brain wiring changes across life. We would simply have to reconstruct the connectome across all of these larval stages or all these life stages, look at the changes and see what's going on. So to do this, we developed a high-throughput serial section electron microscopy pipeline, which allows us to reconstruct the entire brain of a nematode in a few months. So hopefully this video works somewhat well across Zoom. But what you're supposed to see is an electron microscopy volume that consists of about 5,000 images covering an entire C. elegans brain. So here we're just seeing the images at low resolution, but each frame here is actually consists of more than 1,000 megapixels, meaning that we have enough resolution to clearly see individual neurons and synapses. So getting a volume like this involves staining the animals with heavy metals to make the cells and membranes visible to electron microscopy, then cutting the animal into thousands of ultra-thin sections that are imaged one by one, and finally tracing all the neurons and annotating their synapses throughout each of these sections. So here it's just showing all neurons and muscles within the brain colored here by type. So we focused on the brain as it contains the majority of neuron classes, and it's where most decision making happens. And other than having, of course, the anatomy of all the different cells, we also reconstructed the connectivity, so basically labeling every single synapse between every single cell, which it should show in a second. There we go. So basically this way we can reconstruct the connectome of an animal, the brain connectome. So we've done this for 8 isogenic animals across development, which can be seen here on this timeline. And with these data sets, we can examine how the synaptic connectivity and morphology of each neuron change across maturation at the level of an entire brain. So when looking at this data, one thing that was immediately obvious, even just from this visualization, is that the brain is getting bigger without changing in overall shape. We can quantify this by looking at individual cells, such as here is a neuron pair named AIZ left and right. And then we measure the length of each of these neurites, as well as the physical contact area between the neurites. So the total length of all neurites in the brain increase about 5-fold from birth to adulthood. And this growth is proportional such that the physical contact between cell pairs are mostly persistent throughout life. And here the y-axis is simply the percentage of physical contacts between cells that you see across all data sets. So for example, more than 90% of all the physical contacts at birth is present throughout life. So the overall shape and relative size of neurons in the brain is maintained after birth. But in parallel to this neural growth, there's an extensive formation of synapses, which we can see if we just plot the connectomes as network graphs here, where each circle represents a neuron or a cell. There's also muscle cells. And each line represents the synapses between cells. So from birth to adulthood, the total number of synapses increases 6-fold. But this synapse number increase is not simply explained by enlargement of existing structures. As I'll show you, there's actually a profound amount of rewiring going on here at the level of individual cells to the level of the entire brain. So if we look closer at one of these connectomes, make it bigger, we can more clearly see each cell and connections between cells. And here we define a connection as being all the synapses between 2 cells. So some connections consist of more synapses than others and are therefore stronger. So the addition of synapses during maturation creates many new connections, meaning that synapses are added between cells that were not connected before. And the addition of synapses also strengthens existing connections, meaning that the number of synapses per connection increases. So we asked whether this large increase in new connections and strengthening of existing connections happened uniformly throughout the brain. And I'll show you in 3 different contexts that it's not completely uniform. So first, if we look at the creation of new synapses, which is the ones in this plot here, one long-standing hypothesis in developmental neuroscience is that synapse formation is driven by physical contact between 2 cells. So according to this model, neurons sharing a large contact area are more likely to build new connections. So to determine if this was the case, we measured the contact area between every single pair of cells in the brain at birth that did not already have a connection and counted the probability of a connection appearing later in life. And we saw that a large contact was indeed predictive of a new connection forming. So the physical contact formed at birth appeared to create a constant scaffold within which network formation can unfold. And a second form of non-uniform synapse addition was evident if we look between individual neurons. So from birth, it was already evident that some neurons had more connections than others. So we considered the possibility that some neurons were simply more likely to build new synapses, in which case these highly connected neurons would disproportionately make more new synapses compared to other neurons. This idea is also known as the rich get richer. So consistent with this idea, we found that the number of connections that a neuron had at birth correlated with the number of new input connections they received later in life. So here every dot is a single neuron. But surprisingly, neurons with more connections at birth were not more likely to add new output connections. So new connections only converge onto these highly connected neurons. And similarly, neurons with more connection at birth are also more likely to disproportionately strengthen their input connections, but not their output connections. So maturation focuses the flow of information onto the most highly connected neurons at birth. Or said in another way, highly connected neurons become better integrators of information, but not broader communicators of that information. And a third form of non-uniform synapse addition is evident when we looked at the relative strengthening of
Do you have an
neurons.
idea why?
Have an idea why?
Why is that the case? Why you sort of underuse the other neurons and you just keep overusing some of them? Is there a reason we can think of, or?
I mean, there's several ideas, but we really can't tell from our data which might be the case. All we can say is there is this principle, and what is actually causing it is sort of up in the air. Yeah.
I was going to ask how instead of why. So, what is there a mechanistic understanding of growth cone guidance or something that explains some of these things that one can point to?
So many of these changes happen within sort of the stable scaffold of what's already there. Like the structure is sort of already set up. So these neurons, they don't
spine growth
really,
or whatever it is. So something has to happen.
right, actually, and there's minimal spine growth as well. There's some, but the majority of these connections actually happen along neurons that are already next to each other, making contact. Yeah, so it's not clear what's actually causing it. Yeah.
So Daniel, I know we're not supposed to be interrupting you, but one quick question. Your neuron, do you differentiate between excitatory and inhibitory? Because this is sort of a little bit different than what I had learned years ago when I was in Göttingen about development in young chickens. You know,
Yeah. It is a good question, and it's something we've for a long time wanted to be able to answer. But one of the problems with trying to attempt to answer that is that a lot of neurons can be either excitatory or inhibitory, depending. It depends often on the postsynaptic neuron. So the same neuron using the same neurotransmitter can be excitatory at one synapse and inhibitory at another synapse. And so we know the neurotransmitters of pretty much all of these neurons, but we don't know exactly where all the receptors are. So we don't actually know which specific synapses are excitatory and which are inhibitory.
okay, thank you.
Yeah. Okay, moving on. Right, so a third pattern we were looking at is comparing the relative strengthening of a neuron's inputs and outputs. So we correlated the number of synapses for inputs to the same cell, outputs from the same cell, and connection between different cells. And I'm going to show the result in this plot here, where the y-axis, it's a bit scary, the coefficient of variation in synapse number, it simply means how different the synapse number is between two connections. So over time, unsurprisingly, connections to and from different cells diverge in synapse number. The same is the case for inputs to a cell. But interestingly, the relative strength of outputs from a cell are maintained. And this contrast between inputs and outputs, it's mostly clear if you just take the difference between the two, which is shown in the plot here. So the relative strengths of outputs, but not inputs, are maintained across maturation. Or said another way, each cell regulates the strengthening of its own synaptic outputs, but does not dictate the relative strengthening of its inputs. So in summary so far, the maturation of the connectome is not simply uniform, but it follows certain principles that are evident at the level of individual neurons. But these three principles here, they come from overall statistical analysis of anonymous neurons. And one of the advantages of using C. elegans is that we can actually identify the same cells across different individuals. So we can examine exactly which specific connections change. For example, if we just take here a toy example of a circuit consisting of about 6 neurons, we know what the identity of each of these neurons are. So we can directly compare their connectivity across these different animals. So we see that some connections, the black ones here that are present throughout all life stages, we call those, or classify those as stable. Then connections that show a statistically significant increase or decrease in synapse number across development in a stereotyped manner, we classify as developmentally dynamic. So these connections sometimes even form new connections, which is the case for this connection here, or they prune connections away. And then finally, connections that exhibit no consistent trend in their changing synapse number and are not present in every animal, we classify as variable. So looking at these variable connections, we were actually surprised to see that they're fairly common. They consist of almost half, in the adult, they consist of almost half of all the connections, the same as the number of stable connections. These variable connections are typically weak, consisting of fewer synapses than other connections. So if we look at the total number of synapses making up these variable connections, the proportion is smaller, about 15% of all synapses within the brain. But it's still a big chunk, so they cannot simply be ignored. And this degree of variability contrasts with the widely held view that the C. elegans connectome is hardwired. We considered the variability is more prominent during development than in the mature connectome, in which case we would, like by our estimate, we would overestimate the prevalence of variable connections. But if you do a similar analysis where we just consider the different connections between our two adult animals, we get similar numbers. So one thing indicating that these variable connections have some importance is their prevalence. Another is that they're not simply randomly distributed throughout the brain. And we can see that if we look at the connectome and classify each neuron into different cell types. So cell type here being sensor neurons, modulatory neurons, interneurons, and motor neurons. Then if we simply compare the number of variable connections versus non-variable connections for each cell type, we find that variable connections are more prominent among modulatory neurons and least common among motor neurons. This high stereotypy of synapses from motor neurons may reflect a requirement for high fidelity in circuits for motor execution. On the other hand, modulatory neurons, which can release monoamines and neuropeptides by volume release, they may have the weakest requirement for precise positioning of their synapses because they can exert long-range effects. But this non-uniform distribution suggests that variability is in some way regulated and may be functionally important. And this makes sense because even in isogenic populations, behavior is variable. This is evident when giving C. elegans a choice, for example, to crawl or not to crawl towards a certain odor. So you can see that here where each of these squiggly lines is one worm, and an attractive odor has been placed here on the right. And this attractive odor is indicative of food for the animals. So if this video works, over time, the majority of neurons or the majority of worms in this population, they crawl towards this odor in search of food. But there's always a certain proportion of the population that consistently makes another choice and just moves in another direction or ignores the odor. And this behavioral variability is advantageous to the population because if suddenly this attractive odor becomes dangerous to the animal, the entire population would die out if everyone behaved the same way. So behavioral variability is not random, it's highly regulated, and since wiring variability is not random either, it could be a source of behavioral variability. And with this in mind, it also makes sense that motor neurons have the least amount of variability because the worms want there to be variability in the decisions they make, such as should I or should I not crawl towards an odor. But when they made that decision, it should be executed faithfully by the motor neurons. So variable connections are likely important. Similar to how we examined the variable connections, we asked if developmentally dynamic connections, which are the ones that change across maturation, are evenly distributed throughout the connectome. So again, we group cells by their type, this time showed a little bit differently as a network graph, where each arrow indicates all the non-variable connections going from the cells of one type to another. And we found that developmentally dynamic connections are distributed throughout all layers of the network, but most changes are seen at the periphery between sensor neurons, modulatory neurons, motor neurons. And in contrast, the communication within the interneuron layer that may constitute the core decision-making architecture of the brain is remarkably stable. This stability across maturation implies that the central processing unit is robust enough to be used in different contexts. So maturation changes the flow of information from the sensory circuits into the central processor and changes its readout without changing the central processor itself. So we have observed patterns of synaptic chains, which are evident at the individual neuron level, as well as patterns evident at the level of neuron types. And finally, we wanted to ask if the collective set of synaptic changes lead to changes in information processing at the level of the entire brain. So we looked at that in two different ways. We examined first the directionality of information flow. So we classified connections from the sensory to motor layer as feed forward, connections from the motor layer to the sensory layer as feedback, and connections between neurons of the same type as recurrent. And so a network with more feed-forward connections is thought to be more reflexive, with a certain input consistently resulting in the same output. And interestingly, we found that across maturation, stable feed-forward connections were preferentially strengthened more than feedback connections and recurrent connections. And similarly, new connections added during maturation were more likely to be feed-forward, and the connections that were weakened or removed during maturation were more likely to be feedback. So together, this results in the connectome becoming increasingly more feed-forward biased. So one global pattern of brain maturation increases the signal flow from sensation to action, making the brain more reflexive and less reflective with age. And another global pattern of brain maturation was evident when examining the community structure of the brain. So we used weighted stochastic block modeling to group neurons that have similar connectivity into distinct modules. And we found that the connectome becomes more modular across maturation, increasing from 2 or 3 modules at birth to about 6 modules in adult. So synapse formation progressively increases the community structure of the brain as subnetworks for sensory or motor processing gradually emerge with maturation. And interestingly, these subnetworks, they're actually spatially organized within the brain as well, which is somewhat reminiscent of brain lobes in larger animals. So to sum up, we've uncovered principles of synaptic remodeling at multiple levels of the connectome. At one level that applies to individual neurons in the brain, we observe patterns of synaptic remodeling that alter the number and strength of individual connections. And at a second level, we observed synaptic remodeling that differed between cell types. And at its third level, we observed network changes that altered the directionality of information flow and the segregation of information processing throughout the brain. And before I end, I need to stress that a lot of people put a lot of work into this project, and I want to thank everyone involved, including but not limited to my old lab in Toronto with Mei Shen, and my current lab with Arabi Samuel and Jeff Lishman and the Shubit lab, and many others.
Great. Thank you so much.
Thank you. Sorry if I went a bit fast over the last part. I think I wanted to finish on time.
That is okay. Do we have questions from students? If you have a question, you can unmute yourself and go ahead and ask.
Can you hear me?
Yeah.
I just had a quick question about you said you did multiple C. elegans. Were they all under the same conditions? And have you compared like the C. elegans living in like a harsher environment versus those in a more stable environment where not necessarily learning has to occur faster, but that kind of makes sense, sorry.
Yeah, that's a great question. And I'm just writing up a fellowship proposal on that. Because yeah, all of these were done in the same conditions, as close as we could get to same conditions. So there's always going to be like micro environmental changes of worms maybe bumping into each other and stuff that we can't account for. But as far as we could, they're all in the same conditions, as isogenic as we could get them, to try to sort of eliminate that as being the cause of any differences we saw. And because of the low throughput of actually acquiring these connectomes, that's the only thing we've done so far. But we are now attempting to do exactly what you say, take worms that's been in different environments, see how that affects their connectome and their behavior. Yeah, to basically see what do they learn? Like how much of these changes we see are because of genetically programmed things that would happen in every single condition, and how many are because the worms are adapting to their environment.
Thank you.
Do we have another question?
I have a question.
Okay.
There's any student out there that still has a question or not?
I think we had a student question, so you may go ahead.
Okay, so I would like to ask the following thing. If you go to this idea of modularity at the end, you're telling me that each neuron is just involved in a specific task, right? So they're not sharing tasks. Is that an optimal thing, or that's some sort of a waste of resources?
So I don't think that's necessarily the case. So there's a lot of, this method of determining what modules there are forces neurons into a specific module, right? But there's a lot of neurons that are at the edge of what you'd call a model. And there's a lot of, like, even if you look at the two adults, they're not exactly the same, because there's some neurons that sometimes might belong to one module and sometimes to another. So I don't think it's necessarily the case that each neuron does a specific thing. And actually, that question leads up very well to the next talk, because Vlad will show you that in a lot of cases, neurons can actually do a lot of different things. Yeah, so I think that's just sort of an artifact of thinking about it because of the way we've done it. Just trying to show that it seems that there are more modules, but not necessarily that the neurons are forced to be within these modules.
Okay, thank you. Wonderful talk. Really enjoyed it.
Thanks.
Super. Well, I think we'll go ahead and switch now. So now we have Vlad Susoy, who is a postdoctoral fellow. I forgot to mention that these are both researchers from Harvard. And Vlad is going to talk about brain-wide functional organization of a natural multi-step behavior. So when you are ready.
Hi, hello. Can you see my screen now?
Yep.
That's wonderful. First of all, I'd like to thank the organizers for giving me the opportunity to speak about this work. And we are starting with the same model organism, which is C. elegans, and Daniel's given a brilliant introduction into C. elegans connectome. And in this talk, I'll be talking more about the brain function and the functional imaging of the C. elegans brain. So C. elegans, one thing that perhaps should stress one more is that two features of C. elegans, and this is the small nervous system, the small size of the nervous system, and the invariable cellular composition of the nervous system is a huge asset for many areas of neuroscience. And it helps us to answer questions from multi-sense integration to decision-making to representation within the brain. And people have been studying C. elegans neurons for over 3 decades. And at the beginning, especially, people would study a single neuron or groups of neurons. And specifically doing certain tasks. But in the past 5 years, the field has changed since it became possible to look at the every neuron in the brain of C. elegans and see what they're doing, look at the activity. And when people started looking at the brain of C. elegans in this way, they first looked at the immobilized animals, because this is easier to accomplish. So the worms are glued on a slide, And then with the microscope, you can see the fluorescence in neurons, which are indicative of the neurons activity. And what they found was kind of surprising. So first of all, they found that there is quite a bit of activity in the brain of the immobilized worms. But also this activity is very, not to say boring, but it's very low dimensional. Most of the neurons, they participate in coordinated, highly correlated activity, which could be projected on very few principal component axes. And later studies, which were able to look at the freely moving worms with a similar technique, they found that if you allow the worms to move, then the brain activity becomes more diverse, more complex. This is quite a difference from the immobilized worms. And this type of findings have been echoed in the mammalian literature as well. So if when people look at fixed mice, often neurons show correlated activity, large groups of neurons showing correlated activity. But if the mice are allowed to be engaged in tasks and more complex tasks, the brain activity becomes progressively decorrelated. So it seems that the brain-wide activity is a function of the sensory input and also the behavior that the animals allow to display, which is not that surprising. But with that, we also need to keep in mind that most of the behavioral setups are very restrictive for the animals. The animals cannot, or in most cases, it is difficult for the animals to show the full range of behaviors, and as a consequence, it's hard for the brain to show its full range of mechanisms. So we were wondering if we could find a paradigm in which we can look at how the entire brain works during the performance of complete natural behavior. So this is the paradigm that we are using. Here you see two C. elegans worms. They come in two sexes. The bigger worm here is a C. elegans hermaphrodite. They lay eggs. And the smaller worm here is a male worm. And their purpose in life is to mate with the hermaphrodite. To do so, they express this elaborate behavior, which consists of many, many steps, which are guided by the sensory stimuli perceived from the hermaphrodite. So the male finds the hermaphrodite, and then it contacts the hermaphrodite with the ventral side of its tail, which is equipped with many sensory neurons, and then starts pushing backwards, trying to locate the hermaphrodite vulva and insert its mating organs, which are called spicules. And to do this, sometimes the male has to turn around the head of the hermaphrodite, and it expresses many other conserved behavioral so-called motifs. So this is conserved behavioral elements. So as a summary, just summary introduction to the behavior, this behavior is innate, it's social, it's goal-oriented, and the behavioral trajectory of these animals is variable, but it has stereotyped animals, stereotyped elements. Right, and this is just a picture summary of the movie that you just saw. Again, the male shifts between different behavioral motifs. These are stereotyped behavioral components. And one thing which is also crucial for this behavior, or a crucial feature of this behavior, is that the behavior itself is goal directed. So there is a certain goal that the male tries to achieve. And this is very similar to many other behaviors that we see in animals. To perform all these behaviors, the male has a very extensive sensory periphery. And here we are looking at the tail of the male, which is equipped with a number of sensory sensilla. And can you see my pointer?
Yes.
Yes.
Oh, great. So so this green colored structures, singular structures, are called male rays, and the male uses these structures to sense the body of the hermaphrodite, and later we showed that many of them are tuned to different features of the hermaphrodite. There's additional sensory structures here and here, and in total we are talking about more than 50 different sensory neurons. And these sensory neurons, as well as other neurons involved in mating, they are localized in the tail of the male, and in a way they form a separate brain in the posterior part of the worm. And what is interesting is that the number of neurons involved in this posterior brain is very much comparable to the number of neurons in the head of the male. And these neurons involve sensory neurons, interneurons, motor neurons, and a lot of sensorimotor processing, as far as we can tell, happens locally here, although, of course, tail has to coordinate with the head, and this communication between the two brains. But for the purpose of this talk, I'll be talking about the posterior brain only. The connectivity of this brain has been reconstructed previously in 2012. And here we want to provide a functional description of the circuit and see how it works. We could do this with a newly developed tracking microscope system, where we can track moving animals while recording activity of their brains. And on the next slide, I'm going to show you a movie. But before that, just to orient everybody, so here we have hermaphrodite worm. So that's colored in white, and it expresses fluorescent proteins so that we can see the hermaphrodite. And this is the male tail, and it's outlined. These are the sensory rays, and these are individual neurons. And these neurons, they express a calcium indicator, GCAM6, which allows to look at the activity of these neurons, and red fluorescent protein, so that we can see where the neurons are. And this is the movie. So here we are following the tail of the C. elegans male in real time. And we are able to look at the tail as the male performs behaviors. The worms are not restrained, so the male actually exhibits all the normal range of the behaviors it would normally show. And it presses its tail against the hermaphrodite. It looks for the vulva right there. And mostly it pushes backwards. Sometimes it falls off the hermaphrodite completely, and then it needs to find it again. And then when it finds the hermaphrodite, it starts scanning for the vulva. These images, they're actually volumetric, although you can see maximum intensity projection only. And they're recorded at 10 volumes per second, which is more than enough for us to capture all the relevant calcium activity. And here, so the male inserts its spicules into the hermaphrodite, and then you can see that some of the neurons become active, and this is a successful copulation, and then it goes into resting. All right, so alongside the behavior, we quantify multiple features. Alongside the recording from the neurons, we quantify multiple features of the behavior, and this includes the velocity of the male and its tail curvature. And we also map the exact position of the male tail to the hermaphrodite, and we know where exactly the male tail is on the hermaphrodite body. So from experiments like this, we get three types of data. We have the neuronal recordings of neuronal activity, which you can see here at the bottom part of the slide. And we record continuous behavioral data, and we also tabulate discrete behaviors that the males perform, such as searching, turning, scanning, copulation, and resting. And what is really crucial for this data is that we are able to identify most neurons from which we record. So we are talking about 80 to 100 neurons from which we can record, and we can identify about 75 neurons in total. we have performed these experiments and extracted data for multiple males, and this is the ethograms. These are individual behavioral steps that these males performed. And as you can see, the behavioral trajectories of every animal is different. And this is because the behavior is driven by the sensory interaction, by the sensory information, and the sensory information depends on the interactions between both partners. First of all, we want to know if the brain operates in the same way between different animals. And since we have the neuron names for every neuron in every recording, we can address this question by building a computational model, a linear model, to try and decode predict the behavioral features such as velocity, tail curvature, distance of the tail to the hermaphroditic tips, and so on, from the neural activity. So we can build a model from the training data. This is 6 different males and several different continuous behavioral features. And to validate the model, we take a completely new animal, take the neural recordings from this animal, and see if we can predict the behavior of this animal from by using the model built on the training data. And then we can iterate between different data sets to test the model's performance. When we look at how the model performs, we can see that we are actually able to predict multiple features of behavior rather well. And this would include the tail velocity and its curvature, sliding velocity, distance to tips, and to the vulva. And this shows that these features are explicitly represented in the circuit's activity, but also that this representation is highly consistent from animal to animal. So if we one advantage of using linear models is, of course, that they interpret it easily, and we can rank all the neurons based on their contribution to specific behavioral features. And if we take, for example, this neuron, it doesn't have to be this one. We know what most of these neurons are doing from this slide, but if we take this neuron, PVV, which is the top neuron for predicting the distance to the hermaphroditic tips, and if we look at its activity, just by looking, we see that the neuron becomes active specifically when the male swims around the hermaphrodite, and this is highly consistent for within one animal, within instances of this behavior for one animal as well as across animals. And because this neuron appears in all the animals, we can find this neuron in another animal, and we can manipulate it and see what the behavioral effects is. And in fact, if we ablate this neuron in males, we see that the ability to turn around the hermaphrodite is compromised. And what happens here is that PVV is it's an interneuron which collects information from the sensory rays, and then it executes the decision to make a turn. All right, so zooming out a little bit and looking at multiple neurons at the same time, we can aggregate all of our data from multiple animals and from all the neurons by building a consensus correlation matrix. So here's, we're looking at correlations, pairwise correlations between different neuron pairs as well as neurons and behavior. And what we see here is that different neurons, they form groups if we cluster them using hierarchical clustering. And these groups, they roughly correspond to many to the main motifs of the mating behavior, such as turning here. So that's where PVV is, the behavior that I just explained. And the largest cluster here is neurons associated with scanning. And these neurons, they involve sensory neurons, interneurons, motor neurons, and they all become active when the male contacts the hermaphrodite. But of course, what we also can see here is that although hierarchical clustering forces individual neurons to belong to just one group, we also see that neurons can be correlated with other neurons from different groups. And this is, I guess, a common feature among many biological and social networks, that one node in this network can belong to more than one community. One neuron can do multiple things. And to address this, we can use a slightly different approach for clustering, and this is called linked clustering. And what linked clustering allows us to do is to cluster links rather than neurons, such that the groups, neurons can belong to multiple groups. And if we do this, we can see that some of the neuron groups, neurons in some of the groups, such as neurons which associate with scanning or copulation bulb detection, and these are temporarily adjacent behavioral motifs, they often belong to more than one group. And you see a lot of diversity here, just from this picture, different neurons participating in different behavioral motifs. But if we zoom in further onto this purple cloud of neurons, which are all associated with scanning, that's when the male contacts the hermaphrodite and scans for the vulva. You see that, here we have activities of neurons, of 3 neurons from this cluster, and they all align to the onset of different behavioral events, such as turning, bulb detection, copulation, and case contact. And we can see that all of these neurons respond to contact in a very similar way. They R4A becomes active, it's a sensory neuron, PVY, and antineuron also becomes active at contact, and PCA also becomes active at contact. But when we look at different behavioral motifs, for example, turning is also associated with R4A increased activity. But PVY and PCA don't show this. At the same time, PVY has a directional selectivity. It becomes more active when the male starts swimming backwards and less active when the male starts swimming forwards. And this is because PVY actually drives backward swimming. And PCA has a directional selectivity as well, but in a different way. And somehow this neuron is attuned to the speed of relative sliding between the male and the hermaphrodite. And this is because it's a sensory neuron, and it can detect the relative sliding between the male and the hermaphrodite. So there's a lot of diversity in the tuning properties of these neurons, although from the distance they look very similar. And if we plot all the neurons and all the behaviors and look at whether these neurons become more active, and this is in green, or less active, this is in red, at the onset of specific behavioral events, we can see that almost all neurons show unique activity patterns when we look at the full range of behavioral steps that the male is allowed to exhibit. Moreover, even when the neurons are being activated at the same time, we can also see that they often do so in a sequence of activation. For example, in turning, bulb, and copulation, there's a sequence of neural activations which is consistent from animal to animal. And from the ablation studies that we performed and previous ablation studies and optogenetic manipulation studies, we know that these sequences have functional significance. These neurons, they trigger different steps of these behaviors, sub-steps of these behaviors at different points in time. So when the full behavior is considered, nearly every neuron emerges as functionally unique. And I think I'm a little bit maybe over time, but here's one very important point, and that is if we look across different behavioral motifs, what we also see is that functional correlations between neurons, they're not fixed, but they track behavioral dynamics. And here's one example. We have a number of neurons which become active when the male contacts the vulva. And they're all correlated with each other when this happens. But when we look at the activity of the same neurons during a different behavioral motif, some of these neurons become even more active, whereas others become less active. So the correlation, the sign of the correlations between neurons can change depending on the behavioral context. And if we look at the correlations across the brain, and we can do this by partitioning brain-wide correlations for every recording. So this is for every male, and we have 7 males, and we can partition them by the major steps of behavior, such as searching, scanning, copulation, dressing. What we can see is that if you look at the correlation structures, that the columns they look more similar to each other than the rows. So basically, each motif, each of these merge motifs has a more similar correlation structure to the ones from other animals, but the same motif rather than across animals. And another way of showing this is we can arrange these correlation matrices using multidimensional scaling. And we can see that matrices from the same motifs, they tend to be organized together, grouped together. And this applies to neuron correlations, but also neuron to behavior correlations here. And we can track these motif-specific correlations to specific stimulus evoked sensorimotor processes that happen in the brain. And I don't have time to go into detail, but in addition to just doing high-level analysis, we can identify the exact pathways and how they act during different behavioral motifs, during different behavioral modules to control either the same motor neurons or the same behavior. So if the functional correlations change, if the functional correlations can be different depending on behavioral motifs, then the question is how one synaptic wiring diagram supports multiple patterns of functional correlations. And of course, in this case, you would assume that the patterns of functional correlations cannot relate one-to-one to the synaptic connectivity. And this is indeed the case. And to test this, we first, the first analysis that we did would be taking neurons which are correlated and seeing what fraction of them are connected by synapses. And if we do so, we can see that most neurons which are moderately positively correlated, they don't have synaptic connections, neither chemical or electrical. And it is only for the very highly correlated neurons we see that most of them start having synaptic connections. And interestingly, electrical connections are overrepresented in this group. And we can also look at simple correlations between the number of synapses and correlations in activity. And the correlation is there, but it is not very strong, and it is slightly stronger for the electrical synapses than for the chemical synapses. And this is perhaps not surprising because you can imagine that two neurons are functionally correlated without being synaptically connected, and this might happen when they're both engaged by the same by sensory stimuli that co-occur together at the same time. And two neurons can be negatively correlated, not because they have an inhibitory connection, but because they're involved in behavioral steps that happen at different times. The other way of looking at these connectivity patterns and comparing them to the activity patterns is first arrange the neurons by their similarity in activity. And this is using link clustering that I mentioned before. So we have groups of neurons which are both in scanning, turning, searching, and so on. And we can ask if what is the density of connections, synaptic connections, between these groups of neurons and between the groups of neurons. And if we do this analysis, we can see that for the electrical connectivity, the connections between the groups of neurons are more dense than the connections between groups of neurons. Whereas for the chemical synapses, it can be the opposite. You can have very strong synaptic connections between functionally unlinked group of neurons. And one might argue that these patterns of connectivity might be important for coordinating different behavioral motifs. in most cases, we can actually track down these behavioral motifs to track down these interaction patterns to specific neurons and circuits. And one of the circuits is neurons which are involved in valve detection. And these neurons, they have a lot of recurrent connectivity between each other. And some previous studies, and we also showed that these neurons, they are able to excite one another to induce a state when they're all excited. Because they do this, they also need to be under control of another group of neurons, and we identified that these two neurons, which have an extremely strong chemical input onto the valve detecting neurons, and these neurons participate in detecting slide-in. So this is the adjacent behavioral motif to valve detection, and the bomb would be going between these two behavioral motifs many times. What happens here is that the crosstalk between these two groups of neurons enables very robust switching between these adjacent behavioral motifs. And I don't have time to go into any detail here, but the way we determined it is by manipulating the circuit, ablating different neurons and imaging from the remaining neurons in the behaving animals. All right, I guess I skipped to this slide, and this slide shows the multiple sensorimotor pathways that we were able to identify using this approach. And what we've learned from this study is that the brain-wide dynamics in the during this complex multi-step behavior, well, is also complex, but this complexity is driven by the sensory dynamics. It's not an intrinsic property of the brain, but this complexity arises from the complex patterns of interactions between the animal and its environment. And here, one of the other things which is important to mention is that the feedback is very important for this behavior. And this is because the brain, it's not simply passively receives this sense information, but also generates its own experience. So the brain enacts much of the sense information here. And moving on from this, it's been a very, I'll say we are very good now at acquiring more data, but we've been somewhat slow with the data analysis, but now we have a very talented physics student in our lab who's been using machine learning techniques to do the automated posture segmentation and neural tracking. And the challenge here is that in C. elegans, the brain moves all the time. So not only the behavior is dynamic, but the physical appearance of the brain always changes. And that represents a lot of challenges for us. All right, so with that, I'd like to finish, and I'd like to thank many people who have been involved in this project. And first of all, Vivek, who's been brilliant, and he's built the microscope and the tracking system. He's been absolutely crucial for this project, and Ravi for his mentorship throughout this time, and May from University of Toronto, whose lab generated the imaging strain, which has been crucial for this project. And I'd like to thank everybody involved and funding, and thank you for your attention.
Thank you so much. That was a lot of information. Do we have a question from a student?
Come on, students, ask a question before I have to ask a question. I have several questions, but I'd rather wait for you guys first. So let me ask while they think.
All right.
I want to go back to the question I asked on the previous talks about neurons that participate on two modules, basically two different behaviors. are these neurons that are in the middle, they're just sort of transferring information between the two decision-making blocks, or they're actually part of the decision-making? You know, in a sense, or just sort of, is there any way you can figure this out?
I think yes, and in a way, in a network like this, it seems like almost everything is a part of the decision-making. And even when we have two sensory modules interacting with each other, they are also part of the decision-making. In a way, the sensory modules can decide among themselves what information to pay attention to, and then pass down the information to the interneurons or to the property effectors. So I would say the decision-making is very much distributed in the network. This is my view.
Okay, but I agree with you in a sense, but in a sense, basically, you also have some blocks that you say they're associated to different sort of phenotypes or different kinds of behaviors. And the curiosity is that basically in some of these blocks, you may have sort of, you have some master neurons that make sort of the decision, or is all distributed, and then sort of the peripheral neurons in between are sort of just getting inputs and outputs from everything else and sharing, but we still have some major blocks that do whatever. They may be a repressolator like, or they may be a toggle switch or something that's really decision liking.
So I would say that there are very important neurons which drive the behavior. For example, it's a very interesting question. So there are neurons which are necessary for certain behavioral motifs. But I would rather think of them as effectors, although they also integrate information.
Okay.
But I don't think it would be, I can think of any concrete example of a neuron which I would call a decision maker, because the decisions are being made from the sensory neurons down to the motor neurons.
All right, do we have a student question now? You've been thinking and thinking. I have a technical question, just because I am not familiar with how to study C. elegans. How do y'all measure neuronal activity?
I guess I should have spent some time explaining this. But the way it is done is by expressing proteins which are calcium indicators. So these proteins, they can bind to calcium, and they also have fluorescent properties. So when a neuron becomes active, there's a calcium influx in the cell. The proteins, they bind calcium, they become more fluorescent, and this is what we measure. So what we measure is the fluorescence of in these different neurons, which is a proxy for the neural activity.
That's really cool.
There's a question in the chat.
Would you like to unmute and ask yourself, or I can read it from the chat? Okay. The question is, could you elaborate more on how cells' names were identified in the videos? And then there's a second part I'll ask you after.
Right. So the identification of the cells is a very, very important point here. And when we look at these neurons, they're very dense, and it's not trivial. So it took us more than a year to put names on all these neurons. And some of it comes from the stereotyped positions. Many of the neurons, they have either stereotyped positions or stereotyped cell morphologies that we can identify, but many don't. Sometimes we had to find so-called marker strains. This is C. elegans strains that express specific markers in specific neurons, maybe 1 neuron or 5 neurons. And we would do the whole brain imaging with those strains as well to confirm that our identification in the pan-neuronal strain with all the neurons is correct. So it's been a very long process for us. And we are still unable to identify about 30% of that circuit. But that will change because there are new strategies that strains available for neuron identification. There are strains where every neuron is labeled with a different and unique color.
Cool. And the second question is, what do the activities of these cells look like when there is no mating behavior happening? Does this tell you anything about mating specific activity?
So this is a great question, and this is relates to the initial studies on the immobilized elegans, but they saw the brain activity and they thought that this is representative of the fictive motion, fictive behavior. So somehow behavior, so some kind of representation of behavior can be separated from its execution. I think in this case, we definitely show that the representation of behavior cannot be separated from the behavioral performance. The performance of the behavior is what drives activity of these neurons. And of course, we would see activity in some of the neurons, and those are mostly neurons which are not involved in mating. These are neurons which are mostly motor neurons which are involved in swimming, and the males, they swim when mating and when they're not mating. But even the activity of these neurons looks different from when we look at the actual behavior.
Okay, I want to thank both of you for wonderful talks. I think I learned a lot, and I think one of the amazing things is sort of a lot of the things you were showing today that people have been talking about qualitative behavior, they have been able to do a quantitative understand of it and see how much we know in a sort of quantitative way. And I think that's really a transformative change to the way people do this sort of work on this field. So I really enjoy both talks and glad I had a chance of listening to them.
Thank you.
Super. Well, thanks, and we'll see y'all in October.
Bye-bye then.